Hello everyone, this lecture video is all about urea cycle. The urea cycle is a cyclic biochemical pathway in which urea is produced for excretion using ammonium ions and aspartate molecules as nitrogen sources. And then, the urea produced in liver is transported in the blood to the kidneys and eliminated from the body in form of urine. In the pure state of urea, it is a white solid with a melting point of 133 degrees Celsius, and this is very soluble in water, in which you can dissolve 1 gram of urea in 1 milliliters of water, and then it is odorless, colorless, and it has a salty taste. With normal metabolism, an adult excretes about 30 grams of urea daily in their urine. So this is the chemical formula for urea and the chemical structure, and this is the color of the urea. Okay, there are 3 amino acids that are involved as intermediates in the operation of the urea cycle. These acids are arginine, ornithine, and citrulline. So, the most nitrogen-rich compound in this tree is the arginine. It contains 4 nitrogen, 1, 2, 3, and then 4. Unlike with the ornithine, that is only 1, 2 nitrogen. And for citrulline, it is only 1, 2, 3. So, that is why this is the most nitrogen-rich amino acid in this tree. So, next, ammonia, one of the 2 nitrogen-carrying molecules associated with urea cycle operation. It does not enter the cycle directly. Rather, it is converted to carbamoyl phosphate that serve as one of the 2 fuel molecules for the cycle. The other fuel is the aspartate. So, this one, carbamoyl phosphate, will serve as fuel of this reaction. The reactants for carbamoyl phosphate formation are ammonium ions, this NH4 here, from oxidative deamination, and then carbon dioxide, then water, and 2 pieces of ATP molecules. So, upon reaction with each other, we will have the carbamoyl phosphate. The ATP will be converted to 2 pieces of ADP and 1 inorganic phosphate because the 1 phosphate has been attached here in the amide group. This is amide group. Okay. So, this is now the carbamoyl phosphate. And then, the carbamoyl phosphate now can enter the urea cycle. So, steps in urea cycle. First step is the carbamoyl group transfer. This is your ornithine, the amino acid that is an intermediate in this cycle. It will react with the carbamoyl phosphate and it will produce citrulline here. So, this is the carbamoyl phosphate. It will attach on the topmost part, this one. And it will produce an inorganic phosphate. This will be cleaved and phosphate will be released as inorganic phosphate or free phosphate ion. And the next step 2, the citrulline, aspartate condensation. Meaning to say, when you hear the word condensation, they will be combined to each other. So, this is the citrulline and aspartate, and we will consume ATP, 2 pieces of inorganic phosphate for this reaction. This aspartate will attach on this part, and you will now have the Arginino succinate. Next part is the step 3, Arginino succinate cleavage. When we say cleavage, splitting. So, what will happen here? This Arginino succinate will cleave on this part. So, when that happens, you will now have the arginine and fumarate. Then, the step 4 is the urea formation from arginine hydrolysis, meaning to say the arginine will be breaked with the use of water. So, this part here will be hydrolyzed and you will now have the urea. We consume water here for the hydrolysis, breakage using water. And the arginine will now have the product of urea, and then the part here, the ornithine, meaning to say we have regenerated ornithine plus producing the waste product, which is the urea. Let me summarize that for you. Urea cycle, you have the carbamoyl phosphate with production with the help of the carbon dioxide, ammonium ion, ATP, and water. And with the help of the enzyme carbamoyl phosphate synthetase 1, it will produce carbamoyl phosphate here. And then the carbamoyl phosphate, with the help of the reaction with its ornithine, the ornithine molecule, with the help of the enzyme ornithine transcarboxylase, it will produce the citrulline. And the citrulline here, with the help of the enzyme arginosuccinate synthetase and reaction with aspartate, it will produce arginino succinate. Then this arginino succinate, with the help of the enzyme arginino succinase, it will produce arginine and fumarate. Then this arginine here will be hydrolyzed. It will produce the urea and ornithine as regenerating product, and that is with the help of the enzyme arginase, and it will consume water as well. Urea is being commonly used as a fertilizer because it contains lots of nitrogen, so it helps in the nitrogen fixation. The bacteria will degrade that urea and it will produce nitrogen that is helpful in the plants. And urea is also being used in production of synthetic cloths and fibers. So that is all for urea cycle.